Hello, everyone. Okay, respond if you can see me. We'll just wait 1 minute. So I think we're live. Thanks, Megan. I appreciate that. Okay, so everyone's probably a little surprised to see me instead of Adam here. My name is Salma Ahmed. I'm a postdoc in Mala Murthy's lab at Princeton, and I'm substituting for Adam Calhoun, who runs this invertebrate neurobiology seminar series in collaboration with the Worldwide Neuro. A few logistical things before we jump in. If you, I encourage everyone to ask questions. There should be an Ask a Question button at the bottom of your screen. You should click that, write your question, and at the end of the talk, I will ask Ben these questions. I think we can get started from there. I'll be in the chat if anyone has any smaller questions. It's my distinct pleasure and honor to introduce today's speaker, Dr. Benjamin de Bivort, who's an associate professor in the Department of Organismic and Evolutionary Biology at Harvard University. I'm really excited for Ben's talk today, and it's a huge honor for me to introduce him because, as Ben now knows, his work has had a huge impact on my personal understanding of behavioral variability and evolution of behavior and neural circuits. I'm sure today's talk will be enlightening and invigorating. And with that, I will pass the virtual microphone over to Ben. Thanks for being here today.
Yeah, thank you so much, Sama.
All right.
Okay, so it's my great delight to be here to speak with you all and tell you about our work looking at functional and structural loci of individuality in the Drosophila olfactory circuit. And loci of individuality is a term that we've invented, so hopefully I can convince you it's a thing, that refers to sites in the nervous system that are causally determinative of the individual behaviors that a particular animal shows. Like, what are the differences between it and other members of its species that give it its particular specific behavioral bias. And just as a very minor note, I had some struggles with the Wi-Fi this morning. It's been stable for the last hour, so hopefully everything goes smoothly, but there's some minor probability I'll drop out, and hopefully Sama can take care of the rest of the talk for me. Okay, so I'd like to start at the beginning by expressing my solidarity with the Black Lives Matter movement, the protests in support of Black lives. I think it's critical that we keep dialogue going about how we can make STEM a more welcoming place for marginalized people. And in the spirit of thinking about concrete actions, I wanted to mention to you all the ideas that we've been talking about in my lab group around this question. And a specific one that has been intriguing to us is the possibility that graduate programs might include, as part of their formal curriculum, a requirement for students to participate in outreach or diversity and inclusion activities. And realize that none of the graduate programs I know about include such a requirement. And so a question I would put to you all is, do you know of programs, perhaps yours, that have such curricular requirements? And if so, would you mind contacting me either at the email address, which is at the bottom of the screen there, or through Twitter? There's my Twitter handle, it's just my last name. And let me know what's going on in your programs. I'm also interested if you guys have ideas in favor of this idea of requiring outreach as part of a graduate curriculum. Would be interested to hear your thoughts. So hopefully we can have a conversation about that. Okay. I'd like to tell you about the work that we do in the lab, which has to do with variability in behavior across individual animals. And let's see, Sama, can you see my cursor now? Moving around? I'm in a full screen mode where I can't see any responses, but let me just jump out.
Ben,
okay, great.
I can see your cursor, Ben. Thanks.
Okay, thank you. All right, so you can think of examining behavioral diversity and differences in behavior at different levels of organization. So you could look at these different species of flies and ask what accounts for their behavioral differences. And since these are different species of flies over here on the left, your answer is probably that genetic differences between the species account for the average behavioral differences among them. And you go to the other end of the scale and look at the behavior of a single animal and ask what accounts for behavioral differences within its life, such as changes over development or aging or moment-to-moment variation in its choices. So you might call this intra-individual variability, and this interspecies variability. And the main kind of variability we look at is this intermediate scale, which is marked here as intragenotypic variability. And what this refers to are behavioral differences you see among individuals that all have the same genotype. So this is like looking within a species or within a strain where we've removed genetic diversity. And we do a particular version of this analysis where we grow these flies in very similar lab environments. So nominally, these are animals that have had their genotype matched and their environment matched. And so that sort of takes care of the traditional canonical sources of biological variation, which are genes and environment, right? We've matched both nature and nurture in these animals. And yet, as I'll show you, they have very different behavioral profiles from each other. And so the question is, what is varying among these individuals to give rise to those behavioral differences? And one of the predominant explanations is probably stochastic developmental events, whereas the brain is building itself using the developmental program. There's sort of an inherent randomness in that process, so that even when conditions are macroscopically the same, you get slightly different outcomes in the wiring of the brain from animal to animal, and this has consequences for their behavioral biases. Now, that's not the full story, because we have found out that when you perturb specific circuit elements in the brain, you also change the extent of variability, the magnitude of differences between individuals. So there are active processes happening post-developmentally to tune the amount of variability across individuals. And all of these are the sort of factors that we're interested in studying and characterizing with mechanistic detail. Just to motivate this for many of you, hopefully, it's possible you're already studying intragenotypic variability. So if you're a quantitative geneticist and you're used to parsing variants and breaking it down into its genetic, environmental components and their interactions, intragenotypic variability falls into environmental variability because it's not heritable, right? We're looking at differences in behavior that exist when there are no genetic differences. And so there's no mechanism for this variability to be propagated across generations. And so in the definitions that parse variance in this way, any variance which is not heritable is part of the environmental component of variance. And so intragenotypic variability is part of VE. Oops. Sorry, I keep accidentally clicking instead of just dragging my cursor. All right. If you're interested in diseases and understand modeling them in model systems or in cells, the typical way we think of risk factors for disease as affecting disease prevalence is by changing the mean of a phenotypic distribution. All right. So what I mean is, if we imagine that the blue distribution represents the phenotypic distribution of animals that are healthy, if you introduce a risk factor like a genetic variant or something in the environment, you might shift the mean of that distribution in this rightward direction, which in this case represents disease phenotype. And so you get more individuals with the disease because you've shifted the curve to the right. But it's important to recognize that you can also change the abundance of disease by changing the variance of a distribution. So over here on the right, the mean of these two distributions is the same, but there's a greater abundance of disease in the orange group because it has a higher variance, and so it has more individuals out in the phenotypic tails. And if you're somebody interested in doing hypothesis-driven tests around behavior, and you've done a manipulation or a perturbation, and you want to show statistical significance of the effect that you've done, there's a good chance that intragenotypic variability is giving you a hard time because it's increasing the size of the error bars in your estimate of the mean effect of your manipulation. Now, the differences in behavior that we see intragenotypically are not error in sort of the strict sense. They may contribute to error bars, but they are a real biological signal that presumably has mechanistic origins that we can experimentally investigate. And lastly, there's a field of animal personality which looks at behavioral differences among individuals in the natural ecological context, such as boldness and shyness in foraging. And it's generally sort of implicitly assumed that there are genetic differences that account for these personality differences in animals, but that's an empirical claim. And some of our work says that just as big of an effect as genetic variance is intergenotypic variability in accounting for differences in animal personality. Okay, so hopefully you're now convinced that this is an exciting and important topic, and it's possible you've already been studying it. So how we look at it in the lab is through high throughput behavioral analysis. So we've developed a suite of instruments. So here's one for looking at light preference. Here's one for looking sort of at shade, sort of equilibrium light preference. Here's an instrument for looking at temperature preference. And they all have a common format where there's a chamber into which you load a single animal, so one of these modules or one of these tubes. And then depending on where the animal goes with respect to a stimulus, you see an expression of its individual behavioral preference. And because these instruments are all automated, either detecting animals with electronic sensors or with machine vision, and there are multiplexed chambers for housing the animals, we get a lot of throughput here. We can measure the behavioral preferences of many individuals simultaneously and build up the statistics that let us estimate accurately those distributions. So we're not just estimating means, but we're estimating distributions of behavior. And we've been working on this for, about a decade. And over this time have developed assays to test numerous kinds of behaviors with these various rigs. And today I'm going to focus on work examining odor preference in an odor tunnel assay, which I'll show you. And this has been work that's been led by these four gentlemen here. So Danilo is the only team member in the lab currently working on this project, and he's a graduate student. Matt just graduated. He really took the lead in several of the projects I'm going to tell you about. And also accompanying Matt along the journey were postdocs Matt Shergin and Kyle Honinger. Okay, so the instrument with which we measure odor preference is set up as follows. There's a backlight, and on top of it are some linear tunnels shown here with a top-down camera looking at the tunnels and tracking one animal per tunnel. And into these tunnels, we can flow odorized air, and which odor we flow at any particular moment is under the control of a computer that is hooked up to valves where we can control the flow rate and do dilution of these odors and pump them into the linear chambers. And the way they're configured, odors flow in either end and are exhausted out the sides at the middle. And this is done in a laminar flow regime where there's not much mixing of these two odors. And the only sort of mixing really happens by diffusion right at the boundary of those two odors at the middle at that exhaust area. And our computational fluid dynamic simulations tell us that this is a sharp boundary in the odors that the flies will smell. So they'll be in one compartment and it'll be filled with a single odor, and then they hit the midpoint of the arenas and they encounter a boundary where they can now smell the odor that's on the other side and choose to cross over to that side or not as an expression of preference. And generally, when we have two odors flowing in at the same time, we think of this as a two-choice preference assay. So here's a movie, and this is the view from the camera with the tracking overlaid. And the green lines are the flies. And what's going to happen when I play the movie is the flies will be walking around in a pre-odor period where it's just clean air being flowed into the cells from both sides. And then some rectangles will come on telling you that the odors have now been turned on and are flowing into the cells. And you'll see a change in the behavioral responses of the animal. The animal on the far right here with the magenta trail really illustrates the change in response that you see when the odor comes on. So this animal, which had been walking back and forth along the entire length of the tunnels, now encounters the odor boundary and turns around very reliably, staying on one side. And when the odors go off, it resumes running back and forth along the length of the arena. So we get data traces that look like the two I'm showing you at the bottom, which are of two specific individuals. In the movie here, showed you flowing odors just for a few seconds, but typically we flow in the odors for 3 minutes. And what we do is measure the position in the corridor, so that's the y-axis of these two plots, as a function of time, which is the x-axis. And so the top half represents one odorized compartment and the bottom half the other compartment. And we can measure the preference of the individual by saying, how much time does it spend on one half on a reference half? Oh, I think these numbers have gotten reversed. So this should correspond to 0.2, meaning only 20% of the time is the animal in the green zone. And this is 0.52. Oh, sorry. I've realized that this index is goes from -1 to 1. It's calibrated to that scale. So this minus 0.5 corresponds to an animal that's spending about 75% of its time in the magenta side. And 0.2 is an animal that's spending 60-40 with a slight bias for the green side. Yeah, confuse myself on this because sometimes we make it 0 to 1 and sometimes -1 to 1. Okay, so with this instrument and the relatively high throughput we have, I'm going to show you some basic results about idiosyncratic odor preferences. And these are questions that we ask of most of the behaviors that we've studied in the lab because we think that they are important general questions about idiosyncrasy. And the first one of these is the observed distribution of odor preference scores broad in some sense? As you'd expect if individuals have this high diversity of preferences. And the answer is yes. So you can see the blue distribution is broad and has these tails with a small number of individuals, but some that have these really extreme preferences, basically staying on one side the entire duration of those 3 minutes. But it's important to say, is the distribution broad with respect to a null expectation? And in this case, the null model is the distribution of preference scores you would see if all of the individuals had the same true mean preference, but that there was still dispersion in the measurements coming from sampling error. Because we don't get a perfect estimation of individual bias, there's some error. And so even if all the individuals really did have the same preference, we would measure some variance in our observations, and we can estimate that a couple ways. One is using a binomial, one is by looking at the behavior of the animals before the odors come on, and one is using sort of a resampling method. And all of these null model distributions are much narrower than the observed distribution. So this is important statistical evidence that indeed individual behaviors are different animal to animal. It's possible that these odor preferences that we're measuring moment to moment represent sort of quirky momentary moods that the flies are in. Oh, you know, right now I feel like having a chicken salad sandwich instead of a turkey sandwich, but it's not really anything intrinsic to me as an individual. It's just that momentary preference. And so a way of testing this is by measuring the preference of an animal on one day, taking it out of the arena, keeping it maintained in single housing and testing its preference again on a subsequent day. And we see this correlation. It's pretty noisy, but it's certainly statistically significant. And there's a reasonable correlation between the measurement of preference on day 1 and day 2. And we can do this over longer intervals, and we've done it up to a week, and you see a similar correlation. And this tells us that indeed odor preferences are stable properties of each individual. Now, I'm going to, of course, get into circuitry. And so here's the first slide with some of that. Odor preferences are mediated in flies by the binding of odor molecules to olfactory receptors, proteins that are expressed in olfactory receptor neurons. The organization of the insect olfactory system is very similar to the mammalian olfactory system, where all of the olfactory receptor neurons that express a particular olfactory receptor, so here these two have these sort of M-shaped receptors in the cartoon, all of those ORNs, olfactory receptor neurons, project to the same glomerulus, which is a microcircuit in the antenna lobe, now in the brain. And this represents sort of a channel of processing of that kind of information. Projection neurons have their dendrites in typically one glomerulus and carry the information from that channel further into the brain to association and innate response centers. And between the glomeruli of the antenna lobe are local neurons that help shape the transformation of odor representations from olfactory receptor neurons into projection neurons. And these are predominantly inhibitory neurons, though some of them are excitatory. And they're also known to have stochastic morphologies such that every individual has a distinct complement of LNs. So there is our olfactory circuit. And I'll refer back to this in future diagrams, so you don't need to have it memorized just yet. We have found that as in other behaviors we've looked at, the extent of variability is under genetic control. And so in this case, we're comparing the amount of variability in odor preference between wild-type animals in gray and animals with a mutation in the dopamine receptor DOP1R1. And that's a narrower distribution that you can see in this sort of raw kernel density estimate of the data. And then what we have over here is a posterior distribution from a Bayesian model on the effect of having the DOP1R1 mutation on variability. So this density to the left of 0 means that there's a negative effect on variance of having this mutation. And it's something we can complement by feeding the animals L-DOPA, which is a dopamine precursor. So this is telling us that sort of the tone of the dopamine system is one of these determinants of how variable the behavior is across individuals. And this is a hint, this pharmacological rescue, that the activity of neurons matters acutely, not just the genotype of the animals. And indeed, we see that. So here are experiments where we have used transgenic lines to perturb the activity of local neurons. These are those inhibitory neurons that connect between the glomeruli of the antenna lobe and help shape the transformation of the antenna lobe. So either when we activate these neurons with a thermogenetic effector called DTRPA1, or silence them with a thermogenetic effector called Shabiri, we tend to see this effect of lower variability. So these are different LN lines with the different thermogenetic manipulations. And when there is an effect, we see that it is a reduction of the variability of the behavior in these animals. So this is telling us that the endogenous role of activity in local neurons, that is, what are they doing when we just leave them alone and let them fire like normal, is to increase variability, so that when we mess with the neurons, we see a reduction in variability. And this is potentially consistent with those diverse wiring patterns that the LNs have across individuals. They have different wiring patterns. And if this is a particularly variable point in the circuit, when we silence that layer of neurons, it might not be surprising that we see a reduction in variability across individuals. So these are high-level important points about variability. There's one more experiment that we've run in other contexts, which is a confirmation that indeed individual biases are not heritable. So if you take two flies that had the same preference for octanol, one of these odors, and mated them to each other, we would predict that the progeny would not show any particular preference for octanol, that there would not be a heritable bias to the progeny of that cross. We haven't done that experiment for odor preference, but it is one of the experiments we like to do when we study these behaviors. And you may guess what's coming. I'm showing you the result of these experiments for odor preference specifically, but I can populate in the table now back with the full list of behaviors that we've studied and the result of the equivalent experiments for them. And what you can see is broad consistency here, meaning every behavior we've looked at has a broad distribution compared to null models. Every time we've looked at day-to-day persistence, we see that individual biases persist across days. Every time we've run one of these heritability experiments, we've seen that individual biases are not heritable. We've seen that genotype controls the extent of variability, so the breadth of a behavioral distribution. And the state of neural circuits also affects the extent of variability, presumably in ways that we don't fully understand yet, that have to do with control over the signal to noise ratio of the representations of the stimuli that are evoking behaviors here. And there are some empty cells, these white places. These are not inconsistent data. These are just experiments we haven't done. And you can tell which are the harder experiments, the ones over here. And there is sort of one exception that pops out, and this has to do with the stability of individual biases in locomotor handedness. So this is a tendency of individuals to turn left or right that we see in larval flies, so in maggots. The maggots have individual turning biases, but they're not persistent day to day, or at most only weekly so, but we see them as persistent from hour to hour. So that's why this is shaded yellow. And we think that this is because these animals have a developing brain where you're adding in wiring and you're adding in new synapses. And so the particular details of the circuit that matter for individual bias are in flux because this is a more developing brain than in the adults, which were the rest of the experiments. Okay. So I've told you a bit about the determinants of variability as a phenomenon, like what gives rise to more or less differences among individuals. But what I haven't told you about is what predicts a particular individual's biases. Why does a fly, like this fly in the red circle, go away from the light and this fly in the green circle is going toward the light? Or why does one fly really like to hang out in octanol versus methyl cyclohexanol, these odorants? So this is the question we've been looking at for the last couple of years in the lab, and brings in this idea of loci of individuality. So somewhere in the neural circuit that is coordinating these preference behaviors, there are presumably sites or a site where there's a difference among the individuals that accounts for its different preference. And I'm going to introduce some cartoons here that, grossly simplify the brain and what it's doing into the sort of cartoonish feed-forward chain of transformations of stimulus information into motor outputs. Of course, this is ridiculous, but we still think it's a useful framework for thinking about different kinds of configurations of loci of individuality, how we might detect them. So the colors here have to do with how idiosyncratic is the encoding of information at each of these layers. So when we're looking at the stimulus side, we could suppose that sensors at the sensory periphery, like ORNs, are reliable. And so when we give the same stimulus to multiple individuals, that stimulus is represented in the same way across those individuals. Now, that doesn't have to be true. It could be that even at the very sensory periphery, stimuli are represented differently to each individual, and that this accounts for their different behavioral preferences. But for the sake of the argument, let's say that sensors are reliable and that yellow means a consistent representation of information across individuals. When we look at the other end of this cascade, we know that the motor responses of these individuals are different to the same stimulus. So you give two flies octanol, and one of them avoids it, and one of them is attracted to it. So there's some transformation into this blue state, which represents idiosyncratic outputs of the circuit. So there's some transformation where the representation of information in the nervous system goes from being consistent to idiosyncratic. And in this particular cartoon, there's one place where that transition happens, and it happens all at once. And you could call this a focal locus of individuality, like it's all concentrated right there. And this would contrast with distributed loci, where there's a small contribution towards idiosyncrasy at each stage of processing as you go from the sensory side to the motor side. And obviously, the great thing about working in flies is we have genetic access to all the circuit elements, particularly in the olfactory system because it's so well studied, that we can potentially ask this very question. We can go layer by layer through this circuit and say, does information at that layer represent a consistent stimulus across individuals, or does it predict their idiosyncratic individual preferences? And what's really convenient also about the olfactory system is this glomerular organization. So these 50 glomerular channels in the antenna lobe, they contain processes from ORNs, LNs, and PNs. And so if we put our calcium indicators into ORNs and read out their activity in the antenna lobe, we can have a 50-dimensional representation of the encoding of odors by ORNs by looking at the calcium activity in the axon terminals of ORNs. Or we could express our indicators in projection neurons and look at their dendrites. And we can also have a 50-dimensional representation of 50 glomerular channels encoding odors in the PN layer. So we can sort of do direct model comparison between how odors are represented in ORNs and how they're represented in projection neurons, or also LNs, though we haven't done this yet, and thereby look at the representation of information across this cascade in these successive layers and ask, does it predict individual preferences or not? So we basically brought our odor control rig over to the two-photon microscope. We're not doing simultaneous behavior. Instead, what we did was measure behavior in the freely walking animals in those linear chambers and then prep them for imaging afterwards. And we give them a stimulus of 12 successive odors. These are generally alcohols and aldehydes. And they're listed here. They smell floral or sort of pungent or aromatic. Followed by an alternation of octanol and methylcyclohexanol, which are our focal odors for the behavioral experiments. And these are the classic odors that people look at for olfactory learning in flies. And we get volumetric recordings of calcium activity across the antenna lobe. That's shown here. These are different sections. And this is fluorescence that you're seeing from GCAM expressed in projection neurons. So we're looking at the dendrites here. And you can see little lumpy blobs in these images that correspond to the different glomeruli. So from these volumetric recordings, we can do semi-automated segmentation to pull out those glomeruli. And this is where things, of course, get a bit challenging. It's easy to present this cartoon view and say, oh, you know, we can look at the whole representation of odors across the antenna lobe. In practice, we found that we could reliably, automatically segment out from the calcium recordings 5 to 8 glomeruli out of 50, so it's not a huge portion. And some of the challenges here are that the two-photon recordings are noisy. When you do the volume scanning, you don't get the best frame rate. We're only seeing activity, calcium activity in neurons that, or sorry, in glomerular channels that are responsive to the particular odors that that we stimulated with, and you know, some glomeruli are small and some are more variable in their morphology across individuals. So we ended up with a sort of limited view of certain glomeruli that we could reliably segment. And the data comes in this form. So for those 5 glomeruli here, glomeruli have these sort of arbitrary names that tell you about the positions. This would be like dorsal central glomerulus, dorsal lateral glomerulus. We have the peak delta F over F response of those neurons to each of the odorants that we delivered. And as I said, by expressing our GCAMP in different cellular compartments, we can look at the responses either of projection neurons or olfactory receptor neurons. But we get the data formatted in the same way because we're looking at these glomerular channels. To look at how encoding is working statistically, we take that 5 by 12 response matrix, so the glomerulus by odor response matrix, and linearize it. So now we have, linearize it and make it a column in this case. So here, each glomerulus has a block of 12 odors associated with it, and individuals are the different columns of this matrix. And what you can see, sort of the total visual impact of this matrix is some consistency across columns. So like you see a big orange block here, meaning DM2 tends to be quite responsive to multiple odors and across multiple individuals. But you start to see some differences, right? Here is a fly, number 17, which was not particularly responsive, but was less responsive in DM2 than DM1. And that kind of goes against the main trend that we see across individuals. So from this representation of the data, we can embed the entire odor response profile as a 60-dimensional point down into 2 dimensions for visualization using principal components analysis. And here's what we see. So in this plot, each point represents one trial of 12 odors, and a color represents an individual, and a dashed line links the 2 recordings that come from a single individual. And what you can see is that the 2 points associated with each individual and its 2 trials tend to be closer in this principal component space than points chosen from different individuals. So these yellow points are close, these lavender points are close, these red points are relatively close. So there's a separation of individuals in this odor encoding space. It also is true in olfactory receptor neurons. And we can quantify this by comparing the average distance within an individual, so the trial-to-trial distance of odor representation, versus the distance between individuals. So here, this is the distance now in the entire coding space, not in the embedded PC space. So it's taking all the data into account. And what we see is that the distance within individuals here is lower. It's around 2 in this z-scored space compared to the distance among flies, which is higher, around 2.7. And this is true in both ORNs and PNs, so PNs and ORNs. And what that tells us is that individuals are representing odors differently in both of these cellular compartments. So even at the very sensory periphery, individuals represent odors in different ways. But does this individual odor coding predict individual odor preference? You know, it's possible that the representation is variable without variability having any bearing on individual behavior. So we set out to develop some models to predict individual behavioral preference scores using this calcium data as inputs. But you can imagine there are some dimensionality challenges here, right? We have a 60-dimensional representation of odor responses, and we want to use that to predict a single value, which is odor preference. And we don't have that much data. We have something, you know, a few dozens of recordings, which is pretty good, and we're quite proud of having, you know, an N of 50 or 60 calcium recordings from different individuals, because, you know, the field standard can be more like 8. But it's still not enough to do a comprehensive search where all of the 60 dimensions are potential predictors of behavioral bias. We just don't have enough data to fit many model parameters. So what we did was data dimensional reduction first, did principal components analysis to reduce the dimensionality of our data set, and then asked, are the principal component representations of odor responses predictors of individual behaviors? And so here we have that analysis from projection neurons. And what you're seeing is across bootstrapping replicates, a distribution of R² values for models trying to predict behavior from each of the principal components of odor coding variation. And what you can see is that principal component 2 stands out as often across bootstrap replicates having a statistically significant ability to predict behavioral preferences from the values, idiosyncratic values on the second principal component of odor representation. And then this effect completely goes away if you shuffle the data and rerun the analysis. So it's something real about the data, and it's specific to principal component 2. So here's what the corresponding scatter plot between the measured preference of individuals' behavioral preference and the predicted preference is for that model that we found using principal component 2. It's this nice, a pretty respectable correlation. And this is from the training data. So this is the best fit we get. And here's for the test data. So here, this is new data that we didn't use to develop the model. We simply applied principal component 2 as a model to data that we collected in a second round of experiments and found that indeed we could predict individual preferences from those calcium responses in projection neurons. And okay, so that's for projection neurons. What happens when we look at olfactory receptor neurons? This is what we see. None of the principal components are particularly predictive of behavioral bias. This is the best we can do, is an R² of 0.04. So this is telling us that while representations of odors in ORNs are indeed idiosyncratic, they are not determinative, they're not predictors of the behavioral bias of individuals. So this is idiosyncrasy that doesn't play in to behavioral bias, whereas the idiosyncrasy in PNs does seem to affect behavioral bias. So now we can come back to the cartoon view and potentially color in these nodes, but we can't do it quite yet. So we don't yet know what the R² value of 0.2 or 0.3 corresponds to in terms of the color that we should put into projection neurons here. And to figure this out, we have to do a little bit of a statistical power analysis. So hopefully this isn't too dry, and you'll see why this is an important analysis to do. When we have that measure, that 0.2 or 0.3 R² value, the maximum performance of that model at predicting behavior is capped by the noisiness of measuring behavior. It's also capped by the noisiness of measuring the calcium responses. Now, it turns out the calcium responses are more reliable trial to trial than the behavioral responses. But you can imagine that if we could only reliably measure behavior with an R² of 0.5, because there's noise in that measurement, then the model predicting behavior, predicting a single measurement of behavior from a calcium recording, can only do as well as 0.5, right? Because the noise that's in there in the measurement of behavior is also in there when we try to model behavior. So the measurement error of behavior sets an upper bound on the performance of a model trying to predict behavior. And so in this plot, we have, based on simulations, the amount of biological variance that's explained by a model, which has a performance corresponding to the x-axis, given the reliability of the behavioral measurement, which is the y-axis. So that's given as an R value, and then we can plug in the R² from the model. And the question is, how do we estimate the reliability of the behavioral measurement, which is the y-axis? And we can do that by looking at the temporal stability of measurements of behavior. And I showed you a plot of this already. We know that when we measure behavior on day 1 and again on day 2, we see a reasonable R value. In the hands of Matt Churgan in this more recent experiment, it was a bit higher than what I showed you previously.
Ben, I think you cut out for a little bit.
I need to go.
I think you were just saying the new data set that you got has a strong.
Yeah. Okay. Great. Yeah. So the data set's a bit bigger here, better, and what it's telling us with that R value of 0.55 is that this is about the upper bound on model performance. So we can take this measure of the repeatability and therefore implicitly of the noise of the behavior measurement, and we can take our model performance values down here and plot them into this space. And what we see is we're on a contour of around 0.9. What that means is, given that the model has an R of 0.55 and R squareds of 0.22 or 0.3, the model is doing about 90% as well as you could possibly imagine it doing, given the noise of the behavioral measurement. And so that means that the patterns of activity in projection neurons are really telling us quite a lot about individual preferences, about the vast majority of the variance we expect to be able to explain with such a model. So we can color that in, and using 9 or 0.85 as the value and shading the blue that way, we get this very dark blue here. Now, the olfactory receptor neurons did not predict behavior, so we can shade them in as yellow. And so what you see is consistent with that first cartoon view of loci of individuality, that there's an abrupt transition in the representation of information in the nervous system, where suddenly the signal of individual behavioral bias appears. So idiosyncratic calcium activity seems to be driving idiosyncratic behavior in projection neurons, but what underlies that idiosyncratic calcium activity? Can we account for the transformation in the representation of odors that happens from this node to this node? And so a way that we started looking at this, and this is relatively preliminary work, but it's certainly amazing, is by asking, is there a feature of the ORN to PN synapse, something structural, that predicts behavioral bias? And we predict behavioral bias not in patterns of calcium activity, but in patterns of structural features of the circuit. And so we've looked at Bruchpilote, which is a presynaptic scaffolding protein that's involved in binding vesicles and helping them fuse into the presynaptic membrane. So here you see Bruchpilote expression in ORNs. So that's in the axons of ORNs right as they are presynaptic to PNs. This is the antenna lobe, and we can do that same glomerular segmentation and ask, can, with the 50 channels of glomeruli, or the 5 channels we use for the selective analysis, can we predict behavioral bias from the intensity of Bruchpilote staining in these glomeruli? So we did the same kind of dimension reduction to come up with principal components of Bruchpilote variation across individuals and ask, are they predictive of behavioral bias? And indeed they are. So this is not as clean of a prediction as you get from the calcium activity. So we can go back and we can shade in a presynaptic feature of ORNs, which is predictive of individual preference. It's not quite as blue as the pattern of activity in projection neurons because the prediction is not quite as strong. And it's in the shape of a T because Bruchpilote forms these structures called T-bars, which are the docking sites for presynaptic vesicles. And what's also cool about this analysis is we can take our BRP data, which is normalized fluorescent intensity, and apply the model that comes from the calcium activity. So you don't come up with a special model just for BRP to predict data, just apply the model from the calcium activity, and we still get a respectable correlation. It does not reach statistical significance, but it's certainly consistent with the idea that the same glomeruli are important as predictors for behavior from patterns of calcium activity in projection neurons, as for variation in payload expression immediately upstream in the olfactory receptor neurons. And we're taking this forward a bit now using expansion microscopy to get better views of the synaptic puncta, and it allows us to do some measurements that might have a sort of more biophysical interpretation than just total fluorescence. Like we can calculate the density of puncta, so like the number of puncta in the volume of a glomerulus, or median puncta volume, and the ends are much lower because we haven't ramped up the expansion microscopy yet, but consistent with the trend of these models is punctal density in glomeruli as a possible metric. So to summarize and wrap up, the takeaways here are that flies have stable idiosyncratic odor preferences. And they have idiosyncratic odor representations in the first and second order olfactory neurons, which are ORNs and PNs. So the way odors are represented are idiosyncratic and quirky to individuals. Whoops. But the idiosyncratic patterns of projection neuron calcium responses predict behavior, but not ORN calcium responses. And idiosyncratic synaptic density in key glomeruli may underlie the idiosyncratic calcium representations that seem to be predictive of behavioral bias. And so now I'd just like to finish with a slide that acknowledges the other members of the lab and the alums of the lab and our collaborators and the support we've gotten. And I'll jump back here and leave this up and take questions. So thank you very much.
Thank you so much, Ben. That was a really, really fun talk. So I'm going to read you a few questions from the audience.
Should I change views? Like, should I be looking at you guys or?
Oh, you can't see us, right? So you feel free to lower your your screen. Okay, great. So Yunding has the number one question, but it depends on another question. So I'll ask Zatka has this question first. It says, could flies be selecting the side which they were positioned in at the time the odors were first presented? And then they stick around to keep smelling a new odor. Variability in preference to be accounted for by variability in pre-odor position.
Yes, that is a significant component. It's about 10% of the variance of the signal we see in odor preference. So it's a minor component of what's going on, but there does seem to be some like neophobia signal where they're in octanol, and then when they encounter methyl cyclohexanol, they're 10% more likely to, freak out and turn around or something like that. I don't know if it's a startle or if it is really something like a neophobia, but there is a little bit of an effect like that. One of the nice controls we've done that accounts for this that I didn't show you is a yoked control, because what I've done is shown that there's a correlation between our measured preference prior to calcium imaging and the prediction we get from the calcium model. But it's possible that the patterns of calcium activity we're seeing in that subsequent cording don't reflect the sort of abstract preference value that the animal was expressing during the behavioral assay, but instead the specific pattern of stimulation and experience that it had during that behavioral experiment. And so what we did was we took the traces of some arbitrary flies, essentially, and played them back into both compartments. So no matter where the animal was, it got the pattern of odor stimulation that some other fly got during its own preference assay. And then we asked, could we predict the experience of those yoked control flies through the calcium activity of those recordings we took from the yoked animals? And the answer was we couldn't. It was like the ORNs, where the best models still didn't perform very well. So while it does seem like there's this effect of where the animals start and how that is going to vary from trial to trial, it seems like a small component. And in these yoked controls, we don't see an effect.
All right. So I'll ask Yin's question, which is, I think you addressed a little bit of it. So she says, similar to the above question, could it be less of a preference for a specific odor, but more of a response to olfactory novelty or a tendency to explore? Yeah,
I think I covered that. Don't have too much more to say. Maybe something that connects to this and is actually kind of tantalizing, that pink individual from the rightmost column in the movie. When you see it hit the boundary and it turns around and goes back, it doesn't go back to the end of that arena. It goes this sort of distance, right? And it looks like these Philadelphia, these sort of probably central complex mediated exploratory loops, come back to where you started. So maybe they hit the boundary and say, oh, I don't like this. I'm going to, maybe I like where I am. I'm going to try to come back here and see if the odor's gone away or something like that.
Yeah, maybe some checks or something. Okay, so, another question from Munir Oslam Sevic. He says, did you ever see bimodal distributions, especially testing for dope 1R1?
Well, in dope 1R1, we saw lower variability. right? So I would say it's extra unimodal. For the most part, no. We generally see broad unimodal distributions. And we've done a grand analysis looking at the multivariate structure of multiple measures of individuality across many different measures, across like 80 measures. And sort of the most consistent representation is that it's a giant multivariate Gaussian in about 15 independent dimensions. So generally don't see bimodality. In the particular plot, you did see some almost trimodality, where there's sort of central mode, and then there are these really extreme individuals. I think that what you're seeing there is that this is a sensory metric, and we have long tails that are extending past the value of metric and then accumulating up at 0 and 1.
Got it. Okay. Okay. This question is from Aditya Rajagopalan, and they're saying, "Thank you for the great talk. Just to quickly follow up on the DOPR1 results, the lower variance that you see in the mutant, does that persist? Is that over days?"
Yes. Great. Yeah. So we have done the persistence experiment with DOP1R1 animals, and there's a positive correlation. It's a bit lower, and I think that's what you'd expect just from there being less idiosyncrasy, but probably the same amount of measurement noise. So the correlation we expect to drop a bit, and that's what we saw, but it was still, instead of being 0.4, 0.5, it was 3 or 0.4. So it's not that they are anosmic in DOP1R1 or capable of expressing a preference in the assay. They still show these idiosyncratic responses. There's just less of an extent of divergence in it.
Yeah, cool. Okay, this question is from Navin Pokala. It's saying, "Great talk. Do PN or ORN express DOPR? And are any LN, the local neurons, are they dopaminergic?"
Yeah, I think most people would, I don't know that they do. I think they don't, though I don't go too far out on a branch in claiming that. But I think most people think that the relevance of DOP1R1 is through its role in plasticity in the mushroom body. So you have dopamine serving its normal reinforcement role there, and you have receptors on the Kenyan cells, maybe also on mushroom body neurons. I'm not sure. Isama or somebody in this audience surely knows.
Yeah. What's that? If you're in the audience, you know the answer. Feel free to jump in the chat.
Yeah. So that was sort of our first thought, right? And you can imagine that individual experience mediated or channeled through plasticity into changes in the circuits in the mushroom bodies could account for variability, right? Like they may all, be living in the same tube, but maybe one fly got in a fight after smelling something like octanol and formed an association that changed its future behavior. But if you silence mushroom body, you basically see no effect on variability here. So we've done this a few ways, and so we don't think that DOP1R1's effect on variability is mediated through mushroom body. And another structure that does have high DOP1R1 expression is central complex. So it may have something to do with these little searches and the implementation of preference at boundaries and through local path integration. But that's mostly a hypothesis at this point.
Got it. Okay. So we're getting a lot of questions, so I'm going to start being a little picky by picking.
I'm happy to answer more, right? So you can find me on email or on Twitter, and I'm totally happy to talk more.
Great. Okay, so here's $1 million question from Anonymous, which is, what does the second principal component represent?
Yeah, I don't know, like sort of I don't have any cool little, qualitative interpretation, like it falls on a dorsal ventral axis or anything like that. But in terms of these 5 glomeruli, it's heavily loaded for the difference of activity between DC2 and DM2.
Yeah.
It kind of has 0 loadings for most of the other glomeruli. And you can get a statistically significant prediction bias just by inputting the difference the response in those glomeruli.
Mhm.
Just like a really naive model, which is DC2 minus DM2, gives you a statistically significant prediction.
Right.
So specific glomerular channels for this odor pair.
Sure. Okay, here's a question from BFK Lab. It says the group of Bassam Hassan recently showed that the DCN neurons are loci of individuality for attention-like behavior. And individuality lies in the axonal symmetry of the DCN. So their question really goes down to, can you change individuality, I guess? In your paradigm, paradigm. Yeah.
We tried to drive activity in, if I'm getting all my signs right, let's say DC2 minus DM2 is our predictor. We tried to drive activity in PC2 by activating the ORNs that project to DC2 with TRIP, and and so that you play that through in the model, and it makes specific prediction about which way the animals will go, or specific preference for octanol, let's say. And we didn't see an effect. Now, it's possible we're not, the system is nonlinear, of course, and so it's possible that just by providing drive to DC2 through ORNs, we're not really replicating the pattern of calcium activity in PNs that corresponds to principal component 2. And there's probably more sophisticated ways to do that experiment. So, like Sung-Soo Kim, in his work, positioning central complex bump, in his two-photon scanning, he could do crimson excitation in one pass and a recording of activity in the other. And so if you did, during your volume scanning, a specific pattern of activation across potentially all glomeruli, we may be able to sort of induce a more effective PC2 signal. And you could certainly validate the model by applying that PC2 pattern to PNs directly rather than indirectly through ORNs. Haven't done it yet. So that's the kind of experiment we want to do to establish causality, that this is not just a correlation. There's another cool aspect to this, which is we only build models on 5 glomeruli. There are 50. So did we just luck out that the 5 which mattered, or the 2 that mattered, are in the set that we happen to be able to record from? The alternative explanation to being lucky is that there are correlated changes across the antenna lobe, so that seeing DC2 as more active as a predictor of a particular behavioral bias reflects other correlated patterns of idiosyncrasy across the antenna lobe. And we're seeing a certain slice of it in this analysis. So I think that's quite possible, and it would also explain why just poking at DC2 might not be enough to bring it about.
That's right. Cool. Okay, this is from Philip Brand. It says, do you also see strong inter-individual variability with odors that activate labeled lines? It's sort of similar to what he was talking about. Strong variances, like CVA or geosmin. Yeah.
This is what we really wanted to do as sort of the second analysis in this project. And we did CO2 and we did CVA and we did apple cider vinegar, and none of these gave us good calcium responses, so we weren't able to do the calcium recordings. Probably need to find the right collaborator to really do that experiment. But I think it's obviously a question we think is important, and we tried to do. One thing that did work in our hands was odor versus air experiment.
Sure.
And so, like, if maybe a version of what you're asking is, do we see idiosyncrasy when we drive behavior with odors that evoke very strong responses? It's less about the labeled line and about the strength of the response. Then we can look at this by looking at odor versus air responses, because these are aversive odorants when you give them the choice of not going in at all. They're making a choice. This is like a choice between a trash and a gasoline smell. Like, both smell is bad, but you still have a preference where you want to hang out. We found that calcium patterns in ORNs predicted odor versus air idiosyncrasies but not in PNs. So it's a different site for a different kind of behavior. We think ORNs totally make sense because it could be a sensitivity, it could be number of ORNs, it could be the amount of OR expression in the ORNs, generating idiosyncratic sensitivity to odors.
Got it.
Yeah.
Okay. Okay. This
Sorry,
is
that's the answer I have on that.
no, you're fine. Okay, this is related to your first slide, I think, from Yan Wang. It says, regarding outreach as grad requirement, I wonder if the purpose of scientific outreach and that of student coursework are compatible with one another. And then she has a few open questions. Whom does this requirement serve? How is student progress or learning assessed? Would BIPOC students and other marginalized students also have this requirement? Would students be paid? And there's a few more.
Well, yeah. Sorry, is there more?
I guess she wants to, your thoughts on all that.
Yeah. I mean, it's not a very sophisticated idea I have at this point because it's very new. I mean, this is something we talked about in our last lab meeting, and I'm in the mode of trying to get opinions and find out if this has been implemented in there. But some of these ideas you brought up are part of the reason I think this would be a good idea. Like, one, by making it a program requirement, it's going to mean that the white kids do it too, that it's not just the Black and other marginalized groups who are doing the outreach work, because sort of do it by default, and we need to get away from that. And the idea is that by making it a official curricular requirement, yes, people get paid, which is something else we want to do for this work. You'd get, instead of teaching 4 times, you teach 3 times and do outreach for a different semester. You'd get paid to do that. That's the thought that's that we're developing now. Yeah,
I'm excited to see where that goes. Yeah. Okay, this is from. Maybe we'll take one more question. What's your time like, man? I don't want to take.
I mean, I'm open, but
I'm open too. It's totally.
A couple more.
Okay, let's do 2 or 3 more. Okay, so this one is from Arjit Kant Gupta, who says, "Do we see a broad phenotypic distribution for important odors from the perspective of evolution? And if yes, then how is this helping the animal express appropriate behaviors at critical times during its lifetime?"
Yeah. I think this sort of ties to those CVA geosmin questions. We do see idiosyncrasy in the behavior. This was a hypothesis we had kind of early on before we hit all these many different behaviors, was that behaviors in response, or behaviors that are under strong selective pressure out in the wild might be more similar across individuals.
Sure.
And like, who cares if the animals tend to turn left or right, or who cares about octanol versus methylcyclohexanol preference, because they never encounter these odors. So there's not strong selection for the circuits to produce adaptive behaviors in response to these odors. So a way we looked at that early on was by asking, is there individuality in startle responses? Because we think of this as a behavior that is under strong selection, because if you don't jump when the jumping spike comes, or when the dragonfly is coming for you, you're toast. And we found it was on the list, and it had a bunch of yeses, just like all the other behaviors. Startle response was kind of qualitatively similar. Individual flies have individual, like, contrast thresholds, let's say, for visual stimulus, which is driving startle response, or acoustical power thresholds. We used a speaker to get them to jump. And there's actually some work in clonal aphids showing that they have individuality in startle responses. So it's consistent with other insects. And a possible ecological explanation for this is sort of one of the general rationales we're investigating for variability, and is it adaptive or not? And this is the idea of bed hedging. So when the environment is unpredictable and the nature of the selective pressures are changing over time, then a very reasonable evolutionary strategy the animals can employ is to produce progeny that have these different tunings, these different responses, so that no matter what fluctuation comes along in the future, you have at least some progeny who are fit for those circumstances. And so it might actually be the case that the behaviors that are under more pressure are the ones that are more diversified, if there's the right temporal pattern and fluctuation to those pressures in the environment.
So sort of like a population-wide buffering against perturbation.
Yeah, like what matters to evolution, and this is the phrasing people use in bed hedging literature, is not the arithmetic mean of fitness. It's not the like summed average of the number of progeny you have. It's the geometric mean of fitness, meaning it's the product to the nth root. And that's because if you ever go to zero, you're extinct.
Yeah.
Right. And avoiding extinction is what the species have to do. So it's better to have a lower arithmetic mean to gain a higher geometric mean. And that's the bed hedging trade-off.
Cool. Okay, so I'm going to read one more question, then I'll have a question, and then we'll call it. Actually, Ben, do you mind putting in the chat box your Twitter and email, maybe? Because there's like 8 other questions, and maybe people want to follow up. And I encourage everyone to follow up with Ben if your question was not asked. Okay, and the question is from Muhammad Khalaf, and he says, "Thanks for the nice talk. How much is this variability affected by the mushroom body and its learning involved?"
Yeah. Well, so we had this dope 1 hour result, and we thought mushroom body was going to have this big role, but then specific experiments we did there were to silence it with both constitutive effectors and thermogenic effectors.
Yeah.
So PNT and Shabir and TRIP, and we used sort of a suite of different drivers to hit mushroom body, and didn't see much of an effect.
Got it. Okay. So I'll ask the last one. There's a bunch that I had here, but most of them were addressed. And it's about the larval phenotype.
Yeah.
So do you see biases in the larval stage persist to adulthood?
The one time we've tried, so there are old papers that say associated olfactory conditioning done on larvae shows up in adults.
Got it.
It's my understanding that people who've tried to replicate that have not succeeded. And the one time we tried was in looking at the left-right turning tendency of the larva.
Yeah.
When we were measuring the sort of hourly stability of it, and we kept those animals and tested them as adults and did not see anything. It was a relatively underpowered experiment, so we probably would have only been able to see strong correlations.
Yeah.
At this point, we were sort of convinced we weren't going to see anything because even within the larval stages, it's changing hour to hour. So then to expect that you can go through 3 days brain remodeling the adult and see it seemed like a leap.
A little far-fetched.
Yeah. I mean, we really want it to be true because, it's so incredible what happens in terms of remaking the brain to imagine there are these minor details that matter for behavior that persist through that remodeling would just be kind of really cool.
Yeah, it reminded me of this like old paper in moths or butterflies, and they did like fear conditioning in the larval stage, and then it persists into the adult.
No, I think I think it does happen in insects in certain cases. I'm convinced of that. I just don't think we found it yet in flies.
Got it. Cool. Well, thanks so much, Ben, for this wonderful talk. It's really great to chat with you.
Great. Nice to talk to you.
Everyone else, please follow up with Ben online, and Adam will be, I guess, hosting the next one.
Okay. Thanks, everyone.
Bye.
Have a good day.